It is my great pleasure to introduce our speaker for today. So that is Evangelia Christodoulou. She is a postdoc at the DKFZ, or the German Cancer Research Center, in the Department of Data Science and Digital Oncology within the group Validation of Intelligent Systems. And today, Evangelia is going to talk to us about preparing for clinical translation. So these are going to be about guidelines for validating and reporting algorithms performance in biomedical image analysis. It is a very interesting and very pertinent topic with all the upcoming technologies in this field. So we are very, very happy that Evangelia is here to share her talk with us today. So without much ado, I'm actually going to give it to Evangelia to talk to us. I'm going to stop sharing.
Great. Thank you so much for the very kind introduction. I'm very happy to be here today. And I hope that you are able to see my screen now.
Yes, we do see it. It's perfect.
Right. Yeah, so before giving this talk, I try to get an understanding of the background of the people that will be attending my talk. I believe it, even though I'm talking about the medical imaging modality, I can imagine that it's highly relevant for anybody that is working with images. So before going too much into detail, I can just start and can do a brief introduction of my background and how I ended in this lab that I'm currently in and what has sparked my interest in actual proper validation practices when it comes to AI methods. My talk will be focused on two major components of my research these 2 years that I have joined this division. One relates to recommendations with regards to appropriate metric selection for validating algorithms in the biomedical imaging analysis domain. And the second aspect relates to how one should be reporting after having selected the appropriate metrics, how one should be reporting upon an algorithm's performance. And as an introduction to my talk, as I mentioned, I would first like to give you a brief understanding of what motivates me to do research in this certain field. Possibly, many of you might be surprised that a person that has published such a paper with such a title during their PhD is here to talk to you about AI in medicine. So this systematic review that I published during my PhD was focusing on studies that were developing clinical prediction models, which are models that are using tabular, low-dimensional clinical data, and they were performing comparisons between two vaguely defined or broadly defined types of algorithms, machine learning and classical statistical methods, which in most of the times got to be represented by logistic regression. In this paper, we got to observe, on the basis of some risk of bias items that we introduced, and they were focused on validation practices that we got to observe in those papers, included in our quantitative analysis. When those criteria were not met, when those studies were at high risk of bias, we only got to see machine learning methods outperform classical statistical methods. And only when those risk of bias items were at low risk, let's say that they were not met, did we get to, we saw that there is no benefit of outperformance of machine learning opposed to logistic regression methods. This study is highly cited, so it amounts more than 1,300 citations up till now, and it has helped shape certain policies of the EU with regards to validation or reporting of clinical prediction models using machine learning. And let's say that this deep dive into the flaws of studies that are using machine learning algorithms motivated me to explore fields where machine learning or AI, under the general umbrella of AI, let's say, have some benefit, can actually make a clinical impact. And having observed that most FDA-approved AI methods belong to the field of radiology, I was motivated to reach out to a lab that is actually working on AI methods in medical imaging, and also that has a strong focus on proper validation practices. And as many of you might know, commonly people that do not really get to dive very deeply in a machine learning model, they get to see the tip of the iceberg. But there is so much below that that sometimes, and of course at a great expense to the clinical translation of a model, get to be neglected. And some of those aspects would be the dataset sizes that we're using for the development and validation of such models, the annotation, the quality of the annotation, the validation methodology, the choice of the model, the choice of the metric, the reporting practices, the infrastructure that the lab has available, because of course we do know that modern AI methods are quite computationally expensive. But before really delving into the problems, I wanted to make an introduction to the familiarity of the general population, or the awareness of the general population when it comes to risks that come into play when using AI methods. So in a recent UK survey that was organized by the Ada Lovelace Institute and the Alan Turing Institute, a representative sample of 4,000 adults in Britain were asked questions with regards to how they experience AI. And it was quite surprising to me that 56% of them, like more than half of them, considered that the use of AI to predict risk of cancer is quite beneficial. But of course, they were also very well aware of the risks or the pitfalls that one can fall into when relying too heavily on such technologies over professional judgments. And to give you just a vague, or let's say a very general understanding of how the answers of the study participants were in this study. So as I mentioned before, most of them were considering that the assessment of risk of cancer will be beneficial with the use of AI. But they were very heavily concerned on whether we should be using AI for technologies like driverless cars or, and rightly so, targeted political advertising. And of course, at the moment, we're experiencing an era of AI revelations. I would presume that pretty much all of us are using ChatGPT or other generative AI chatbots on a daily basis. We also have in the medical imaging or the imaging in general domain, the segment anything model. There is a huge success from DeepMind's side. So the AlphaFold algorithm is able to predict using deep learning protein structures. So this has helped science to a great extent. And when focusing to biomedical imaging, there is a wide variety of AI methods that go to get FDA approval, and most of them are around the field of brain imaging or detection of diabetic retinopathy. And currently, possibly many of you are aware that we are in this era of the overhyped foundation models. And there is also one foundation model that is holding a strong promise for generalizable disease detection from retinal images. However, there are also a lot of failures of AI, and apart from the racial biases or a lot of, let's say, risks that AI models are getting to impose on the health of a patient. We also are experiencing currently that a lot of early adopters of LLM-enabled medical devices are using those devices at their patients' risks. These devices have zero regulatory approval, and according to the EU AI Act, those are high risk systems. They have no resilience regarding errors, inconsistencies, and they are offering hallucinations. They are not GDPR compliant. They are not trained on copyrighted. They are trained on copyrighted material, and they do not provide us with any consistent performance for their intended purpose. And this means that validation is highly challenging, and proper validation is highly crucial. This is something that gets to be acknowledged, but not only important so-called stakeholders in the AI domain or in the major startups that are developing popular AI methods, but even back in the day, Peter Drucker, who was a person that has set philosophical and practical foundations of modern management theory, they would say things like, if you can measure something, you cannot improve it. And this leads like a proper bridge for the introduction to my first topic of my talk today, which will be centered around the selection of the appropriate metric for validation of medical imaging algorithms. And this is quite an important question because at the moment, there is a wide variety of available metrics. A lot of them have their positive aspects, a lot of them have their negative aspects. Some of them are designed for a specific purpose, but it's most often the case that we do not have so much clarity with regards to what one ought to select for their own biomedical problem. And in previous research in my lab, we went around to, let's say, the community, where people were participating in these biomedical challenges, where I presume you might know that a new algorithm gets to be introduced, and the winner of those challenges gets to be granted with a massive amount of money. So there is a lot of competition, and people are, of course, very motivated to win such challenges. So when those people were asked questions on how they are selecting the metrics when they're participating in such challenges, most of the times the question was that they just select what is standard for the community, whatever is widely used or whatever other people in the field have habitually been using. And as you can see here, for segmentation tasks, most survey participants would select dissimilarity coefficient or Hausdorff distance. But let's have a look whether this is a good idea later. Also, when it came to classification, most of the participants would be using accuracy or area under the ROC curve. And in one of those very popular challenge platforms that I presume many of you know, Kaggle, it seems that the community is not even motivated to go beyond the custom metrics. So as you can see here, when you're competing for a challenge, or when you're participating in a challenge and you don't use the custom pricing plan, you are only able to use the custom evaluation metric. So people are not strongly motivated to actually use metrics that are fit to purpose, because I would presume that also the community is not very well aware of which metric is fit to purpose. And going around the idea of why we need metrics that go beyond the standards, let us have an intuitive example of having a data set where we would want to develop a model on that would be in a position to classify emails into spam versus no spam. So you can imagine that the data set looks like this. So we only have spam emails in this data set apart from 3 no spam emails. And then we train a model on the basis of this data set and we get those predictions. As you can see here, all emails have been classified to be no spam from the algorithm. And in this case, we have the so-called strong class imbalance in the original data set. And if we were to use a single metric like accuracy, we would be thinking that, oh, okay, our algorithm performs very well. We have a 97% accuracy. But accuracy is a very flawed metric because it learns from the majority class. So it would always output the minority as being part of, predict the minority class to be part of, to be the majority class, let's say the non-spam emails to be spam emails. And if we were to select another metric that takes into consideration class imbalances, like the balanced accuracy, we would see a completely different picture. As you can see here, we drop in performance massively. But is balanced accuracy a metric that we should always be using? Is it something that we should be recommending in every case? So imagine that we have a confusion matrix like this. So we have true positives and false negatives, which are severely underrepresented, and a lot of false positives in our that got to be derived from our algorithm. So if we were to use accuracy, of course, as I mentioned before, we will have an inflated prediction, inflated understanding of the predictive performance of our model. Balanced accuracy as well is, in this case, very inflated and doesn't really reflect upon the fact that the algorithm is not predicting well. And if we were to use positive predictive values instead, since it's a metric that has the false positives in its denominator, we would get to see a completely different picture. And similar to Mathur's correlation coefficient, the metric is actually showing that our model is not performing well. Now, let's imagine that we have another type of pitfall when it comes to selecting metrics. So in this case, we have an image, a reference image from colonoscopy. And as you can see here in this colonoscopy image, we have 3 objects, right? We have a larger polyp, potentially polyp, at least in my understanding, and 2 smaller objects that are, we do not have any understanding of how they will develop or evolve over time. So imagine that now we're using 2 algorithms, and the point here would be to detect all 3 objects in the reference image. So as you can see here in this example, algorithm 1 is only in the position to detect one of the 3 objects, so the larger object, and algorithm 2 succeeds in detecting all 3 objects. If we were to use a metric like the dissimilarity coefficient, we would be misled to believe that the algorithm 1 is performing better than the algorithm 2. And this indicates that the die-score is not an appropriate metric. And if we get to think also of the clinical consequences of this selection of our own metric, imagine that those 2 objects that we didn't get to detect with algorithm 1 would later on develop into malignancies. So it's extremely important to be very much aware of the fact that we want to detect as many objects as possible. So in this series of, in this publication that you can see here, we go to uncover a wide variety of limitations or pitfalls that come alongside with the selection of a wrong metric. But of course, uncovering such problems is very good, but solving such problems is even better. And that motivated us to set up the so-called Metrics Reloaded consortium that is somehow a wordplay to Matrix Reloaded. And our intention is to come up with an as comprehensive as possible set of recommendations for metrics for many driving biomedical problems. So to do that, we initiated a Delphi process that was based on a kickoff workshop that was held in 2020. And we wanted to develop a multi-stage Delphi process for consensus building. And of course, the intention was to reflect as broad a range of imaging domains as possible and expand the available expertise. So the number of consortium members gradually increased from the initial 30 to 73. And our intention was to include members that would provide a wide range of expertise, ranging from biology, medicine, epidemiology, biomedical imaging analysis, all the way to statistics, mathematics, and computer science. So as you can see here in this figure, we just wanted to depict how these questionnaires or these recommendations got to be compiled in an iterative manner. So we would send out the questionnaire, the panel would respond, and then we would get more feedback, refine the recommendations and so forth. And this took a total of 2.5 years of research. And of course, we submitted this paper for publication in February 2023, and it took the reviewers approximately 1 year to go through this manuscript because it's quite large, as you can possibly imagine. So figuratively, we are starting from the driving biomedical problem. Let's say we have 4 different problems, right? And through the so-called problem fingerprinting, we are trying to map the problem categories. So we're starting from the biomedical interest to the problem category. And this is happening, or this is getting to be achieved by answering multiple, asking people multiple questions, and those people will be answering these questions alongside the framework. And then in a structured manner, the relevant problem properties are being captured. And then for every relevant problem property or every set of relevant problem properties, we go to compile multiple metrics, so a pool of metrics for every problem. And in the end, when mapping the metrics to the problem, one got to finally get a full set of metrics for each problem. How did this problem fingerprinting look like? I can give you possibly some examples here. So when people got to identify the type of problem that they had, they got to answer certain questions. So those questions would relate to whether for their problem there was a particular importance of structure boundaries, whether there was a particular importance of the structure center, whether there were small sizes of structures relative to pixel size, whether there was class imbalance available present in the data set, and whether test cases were independent or non-independent from the data that were used for training. And in this manner, our intention was to compile such recommendations that would be able to generalize across domains. So as you can see here, we can do classification on different scales and use similar, if not the same, metrics for different tasks. For example, on the y-axis of this figure, you can see the multiple classification scales. If we are to do classification at a whole image level, we have image level classification. When such a classification takes place at a pixel level, we got semantic segmentation. When we want to classify at an object level, we call this problem as object detection. And when there is a combination of object and pixel level, we're talking about instance segmentation. And as you can see here, we have some examples of the multiple levels of granularities and the modalities that one can consider using our recommendations. In the end, the framework that we go to compile looks somehow like this. I think at a first glance, it's quite intimidating, it's quite complex. There are multiple driving problems that got to be considered. And the different colors of the arrows indicate the problem category. So the blue color indicates image level classification. The yellow color goes for semantic segmentation. The green arrow is for object detection, and the purple arrow is for instance segmentation. And let's say that in our case, we have a problem at hand that relates to image level classification. So we are starting from the problem fingerprinting, as I mentioned before. We're capturing in a structured manner the properties of our problem. And then we move on to the specific recommendations that relate to our problem. So whether we have a multi-class problem for the image level classification, and then we have a variety of candidates, and in the end, we end up with a final set of metrics that we would apply for our problem. And if we were to want to observe a more simplified version of our problem, then you would only focus on the yellow arrow. And thus, as you can see here, the metric candidates are much more simple. And we went around on Twitter also, and we tried to focus on common biomedical use cases. So we have specific recommendations that would fall under certain very commonly encountered biomedical problems. So if people feel very much confused by the framework, they can use those recommendations. And to make it even more accessible to the community, we compiled the so-called metric cheat sheets. And as you can see here, for each metric, we can provide the functional form. An intuitive understanding of this functional form looks like the value range, the definition of this metric, relevant pitfalls, the cardinalities, the type of biomedical problem that this metric is getting to be recommended for, and more general recommendations. You can always access those cheat sheets when going in our when accessing our manuscript. And as a further attempt to make it accessible to the community, because there are a lot of metrics that are heavily correlated or can be computed from one another, we also compiled this quasi heat map of the metrics. And as you can see here, the green one, the green dot boxes indicate that two metrics are exactly synonym. The blue boxes that they are mutually computable from each other, and there are also other notable relationships to take into consideration. We also, because there are multiple packages and from scikit-learn some metrics, would not have the same output that we would compute through our recommendations. We also put forward a standard implementation, and we also introduced a toolkit where a user, as you can see here, I do not have to go too much into detail because I think I'm falling short on time and I have much more things to talk about. But let's say that one person, one user, one researcher would be guided step by step in the selection of their metric without having to read the possibly complicated framework from our publication. And now another topic that I would like to be touching upon is the reporting of these metrics, because up until now I've been talking about the metrics that one should be reporting, using to report model performance on. But sadly, even selecting the appropriate metrics is not enough in order to have a good understanding on whether our algorithm is ready for clinical translation, right? So another project that I've been working on, and I have been receiving support from Annika Ranke, who is the lead author in the Metrics Reloaded paper, is titled Confidence Intervals Uncovered, Are We Ready for Real-World Medical Imaging AI? This is a project that has been jointly supervised by Guy Alvaro and Olivier Colliot, and it has been early accepted in MICAI 2024. And the main motivation for this project was that we commonly encounter results tables that look like this in publications. So we have multiple methods being compared against each other, and we have one proposed method that has a minimal increase in predictive performance compared to its benchmarks. It gets to be added in a boldface number, and then the authors make strong claims that their method is offering an important or substantial improvement over the state of the art. And alongside with the recommendations of FDA, which strongly recommends proper statistical practices and quantification of uncertainties of ML model performance, we decided to explore the following questions. So we focused on the papers that were published on the MICAI conference in 2023, with a specific focus on segmentation papers, since this is the most commonly encountered type of biomedical problem in MICAI. And we performed performance variability related data extraction. And we had three major questions. One was to explore the common practice with regards to reporting a variability in the medical imaging analysis community. The second question was to figure out when these variability parameters were missing in the papers that we screened, that we extracted data from. How can we estimate those variability parameters on the basis of the provided data? And the third question related to how the confidence interval widths, I would presume that many of you are aware of the notion of confidence intervals. So the width would be the difference between the lower and the upper and lower bound, I'm sorry. And how do these widths, so the wider confidence intervals we have, the more stable the model is, the model performance is, and the narrower we do confidence interval we have, sorry, the other way around. The narrower confidence interval width we have, so it's the heat probably, the more stable, the more of an indication we have that our model is performing in a stable manner. And we just wanted to explore how these weights would compare to the claimed improvements in model performance. So from when it came to research question 1, the common practice results, out of the 221 papers that we screened, only less than 50% of them were reporting variability in model performance in any way. When it came to the reporting of confidence intervals, we only got to observe one paper reporting model performance using confidence intervals. And out of those papers that were reporting on variability, standard deviation was reported in less than 50% of them. And out of those papers that actually standard deviation was reported, more than 50% of them were computing the standard deviation in a manner that appeared to us to be unclear. So our idea was to focus on those papers that were not reporting on any variability parameters and recover those variability parameters from the available data. And as I mentioned before, the narrow confidence intervals, so small standard deviations, are desired when it comes to clinical translation. So using available data from the medical segmentation decathlon challenge, we developed a model that would approximate standard deviation, assuming that there is this parabolic or second-order polynomial relationship in this figure here, and this is how we expressed standard deviation as a function of mean die-score. And when externally validating this model using data from external, from 56 past medical segmentation challenges, we go to observe that our model, our approximation model behaves quite reliably. And when it came to the claims that relate to outperformance opposed to the confidence intervals widths, here you can see a graph that only picks every 7th of the papers that we include in the quantitative analysis in our study. And the green bar relates to the CI, the confidence interval for the first rank. The vertical green line relates to, so we only focused on the 2 top-ranked methods per paper. And when you see a red color, it means it relates to the mean die-score of the second rank. And that second rank, when it is included within the confidence interval of the first ranked method, it gets to be marked in red. And we go to observe that these red bars were present in 64.9% of the papers that we analyzed. So it's quite surprising, or maybe to many people not so surprising, to realize that the 2 ranks are unstable in more than 60% of the papers that we analyzed from last year's MICAI conference. So because I know that I'm running short on time, and we will definitely have some questions, the take-home messages of my talk would be that the future of medical AI is based on interdisciplinary collaborations, and for proper validation in AI, one ought to define their prediction problem appropriately, choose the right metrics, and report on model uncertainty. And as you can read, we should also be aware of all the pitfalls that can occur at any stage of the pipeline, and also consider further aspects that can impact the generalizability robustness of our AI application. And with that, I would like to conclude my talk. I'm sorry if I left only very little time for questions, but I'm very happy to be reached out on via email or on Twitter whenever you want to ask me any further questions. Thank you very much for your time.